Welcome to this lecture on animal biotechnology. Today, we are going to explore a truly revolutionary concept known as the production of pharmaceuticals through animals. Imagine if, instead of building massive, multi-million-dollar steel factories to manufacture life-saving medicines, we could simply rely on a healthy, grazing goat to produce that exact same medicine in its milk. This is not science fiction. It is a highly efficient reality that is transforming modern medicine. Let us dive in and understand how this incredible process works. Now that we have introduced the idea of animals as living factories, let us look at what this actually means. This field is known as farming, which is a clever combination of the words farming and pharmaceuticals. Traditionally, complex medicines are produced drop by drop in artificial cells within expensive industrial bioreactors. Farming flips this entirely. By using genetically engineered animals, we turn them into natural biological factories. We can harvest massive amounts of therapeutic proteins simply by milking the animal. This approach is not only much faster, but also significantly cheaper than maintaining artificial factories, allowing us to produce life-saving treatments at a fraction of the cost. To understand how these animals produce medicine, we must look at the genetic mechanism behind it. In animal biotechnology, we create what are called transgenic animals. This means a foreign human gene responsible for a specific therapeutic protein is deliberately inserted into the animal's genome. But we do not want this protein produced everywhere in the animal's body. To control this, scientists attach the human gene to a specific promoter sequence, such as the beta-casein promoter. This acts like a targeted switch, ensuring the gene is only turned on in the mammary glands. As a result, the therapeutic protein is secreted exclusively in the milk, leaving the rest of the animal completely healthy and unaffected. This targeted genetic approach leads us to an important question. Why go through the trouble of using animals when we already use simple bacteria like E. coli to make medicines like insulin? The answer lies in biological complexity. Bacteria are simple organisms. They lack the machinery to perform post-translational modifications, which are complex chemical tweaks, like adding sugar molecules to proteins. If a complex human protein is made in bacteria, it often comes out misfolded and completely useless to the human body. While mammalian cell cultures can do this folding, they are incredibly expensive and yield very little product. Transgenic animals solve both problems perfectly, providing the exact biological machinery needed for correct folding while producing massive, commercially viable quantities. Before we move on to the mathematics of this process, let us visualize the entire farming workflow with this diagram. On the left, you can see the human therapeutic gene being combined with the mammary gland promoter. This combined genetic material is then introduced into an animal embryo. The embryo develops into a transgenic goat, which you see at the top right. When the goat matures, it produces milk containing the human protein. This milk is collected in a bucket and then passed through a purification column, shown as the cylindrical filter. Finally, the purified pharmaceutical is bottled and ready for human use. This diagram beautifully summarizes how we go from a single gene to a bottled, life-saving medicine. Now that we understand the biological process shown in the diagram, let us look at how biotechnologists calculate the efficiency of this system. To determine if this process is financially viable, we use the pharmaceutical yield equation. The formula is Y equals V times C times E. Let us break down every symbol. Y represents the total usable yield of the pharmaceutical protein, measured in grams per year. V stands for the total volume of milk produced by the animal, measured in liters. C is the concentration of the human protein in that milk, measured in grams per liter. Finally, E represents the purification efficiency, which is the percentage of protein that survives extraction. If the volume or concentration increases, our total yield naturally goes up. However, if our purification efficiency drops due to poor filtering, the final yield will decrease significantly, even if the animal produces a lot of raw protein. Before we move on to scaling up, let us see this yield formula in action with a concrete example. We are looking at a transgenic goat that produces human antithrombin, a vital blood-thinning protein. We are given that the goat produces 800 liters of milk per year with a protein concentration of 2.5 grams per liter. The purification efficiency is 80% or 0.80. We need to calculate the total usable yield. First, we find the raw yield by multiplying the volume by the concentration, giving us 2,000 grams of unpurified protein. Next, we account for the inevitable losses during filtering by multiplying those 2,000 grams by the 0.80 efficiency. This gives us a final usable yield of 1,600 grams of antithrombin per year from just one goat. Now that we have calculated the yield for a single animal, this brings us to the challenge of scaling up production for an entire population. To know how much medicine to make, we first need to calculate the total global demand. The formula for this is D total equals P times D. Let us define these variables clearly. D total represents the total global demand for the pharmaceutical, measured in grams. P stands for the total number of patients worldwide who suffer from the specific disorder. The variable lowercase d represents the annual dose required per patient, also measured in grams. The physical relationship here is straightforward. If a disease spreads and the number of patients increases, or if the required annual dose goes up, the total global demand will increase proportionally, requiring us to produce much more medicine. With our demand formula established, let us apply it to a real-world scenario. Imagine a rare genetic disorder that affects 5,000 patients worldwide. Each of these patients requires 12 grams of a specific therapeutic enzyme every single year to survive. We're asked to find the total amount of this enzyme that must be produced globally. We use our formula, where total demand equals the number of patients multiplied by the dose per patient. We substitute 5,000 for the number of patients and 12 for the dose. Multiplying these together, we find that the pharmaceutical company must produce a total of 60,000 grams of the purified enzyme every year to treat everyone suffering from this disease. Now that we know the total global demand from our previous calculation, we need to figure out how many animals are required to meet it. This brings us to our final formula, the required herd size equation. The formula is N equals D total divided by Y sheep. Let us explain what these terms mean. N represents the total number of transgenic animals, in this case sheep, needed for the herd. D total is the total global demand we just discussed, measured in grams. Y sheep represents the usable yield produced by a single sheep per year. Physically, if the global demand increases, we will need a larger herd. Conversely, if we can genetically improve the sheep so that the yield per animal increases, the total number of animals required will decrease, making the farm smaller and more efficient. Before we can calculate the final herd size, we must first determine the usable yield from a single transgenic sheep. We are given that each sheep produces 400 liters of milk annually. The enzyme concentration is 3.0 grams per liter, and the purification efficiency is 75%, or 0.75. We are asked to find the usable yield per sheep. We use our standard yield formula, multiplying volume, concentration, and efficiency. First, multiplying the 400 liters by the 3 grams per liter gives us a raw yield of 1,200 grams. Then, we multiply this raw yield by the 0.75 efficiency. This tells us that one single sheep provides 900 grams of usable, life-saving medicine every year. We finally have all the pieces needed to determine our required herd size. We know from our earlier steps that the total global demand is 60,000 grams, and the usable yield from a single sheep is 900 grams. We are asked to find the total number of sheep required. We take the total demand and divide it by the yield per sheep. Dividing 60,000 by 900 gives us 66.67. Because we cannot have a fraction of a living sheep, we must round up to ensure we produce enough medicine to meet the demand. Therefore, the company will need a herd of exactly 67 transgenic sheep. This beautifully illustrates how a relatively small farm can replace massive industrial plants to supply the entire world. We have covered a lot of ground today, so let us review the key takeaways from our lecture on animal biotechnology. First, farming uses transgenic animals as living bioreactors to produce complex human proteins in their milk, overcoming the severe limitations of bacterial factories. Second, we learned how to calculate the usable yield by factoring in milk volume, protein concentration, and purification efficiency. Finally, we saw how scaling up these calculations allows us to determine the exact herd size needed to treat a global patient population. You now have a solid understanding of how genetically modified animals are providing sustainable, cost-effective treatments for rare diseases. Great job following along with the mathematics and the biology.